Which of the following statements about CATH-Gene3D and HAMAP databases is incorrect regarding its features? A. CATH-Gene3D describes protein families and domain architectures in complete genomes. B. In CATH-Gene3D the functional annotation is provided to proteins from single resource. C. HAMAP profiles are manually created by expert curators. They identify proteins that are part of well-conserved bacterial archaeal and plastid encoded proteins families or subfamilies. D. HAMAP stands for high-quality automated and manual annotation of microbial proteomes. The correct answer is: in CATH-Gene3D the functional annotation is provided to proteins from single resource. Which of the following statements about COG is incorrect regarding its features? A. Currently there are 4,873 clusters in the COG databases derived from unicellular organisms. B. It is constructed by comparing protein sequences encoded in 43 completely sequenced genomes which are mainly from prokaryotes representing 30 major phylogenetic lineages. C. The interface for sequence searching in the COG database is the CONITER program which is based on gapped BLAST. D. It is a protein family database based on structural classification. The correct answer is: it is a protein family database based on structural classification. Which of the following statements about PRINTS and ProDom databases is incorrect regarding its features? A. PRINTS is a compendium of protein fingerprints. B. Usually the motifs do not overlap but are separated along a sequence though they may be contiguous in 3D space. C. Current versions of ProDom are built using a novel procedure based on recursive BLAST searches. D. ProDom domain database consists of an automatic compilation of homologous domains. The correct answer is: current versions of ProDom are built using a novel procedure based on recursive BLAST searches. Which of the following statements about InterPro is incorrect regarding its features? A. Protein relatedness is defined by the p-values from the BLAST alignments. B. The most closely related sequences are grouped into the lowest level clusters. C. More distant protein groups are merged into higher levels of clusters. D. The outcome of this cluster merging is a tree-like structure of functional categories. The correct answer is: protein relatedness is defined by the p-values from the BLAST alignments. Which of the following statements about SCOP is incorrect regarding its features? A. Proteins with the same shapes but having little sequence or functional similarity are placed in different superfamilies and are assumed to have only a very distant common ancestor. B. Proteins having the same shape and some similarity of sequence and or function are placed in families and are assumed to have a closer common ancestor. C. SCOP was created in 1994 in the Centre of Protein Engineering and the University College London. D. It aims to determine the evolutionary relationship between proteins. The correct answer is: SCOP was created in 1994 in the Centre of Protein Engineering and the University College London. Which of the following is not a member database of InterPro? A. SCOP. B. HAMAP. C. PANTHER. D. Pfam. The correct answer is: SCOP. Which of the following statements about SUPERFAMILY database is incorrect regarding its features? A. Sequences can be submitted raw or FASTA format. B. Sequences must be submitted in FASTA format only. C. It searches the database using a superfamily family or species name plus a sequence SCOP PDB or HUM IDS. D. It has generated Go annotations for evolutionarily closed domains and distant domains. The correct answer is: sequences must be submitted in FASTA format only. Which of the following statements about PANTHER and TIGRFAMs databases is incorrect regarding its features? A. TIGRFAMs provides a tool for identifying functionally related proteins based on sequence homology. B. TIGRFAMs is a collection of protein families featuring curated multiple sequence alignments, hidden Markov models, HAMs, and annotation. C. Hidden Markov models, HAMs, are not used in PANTHER. D. PANTHER is a large collection of protein families that have been subdivided into functionally related subfamilies using human expertise. The correct answer is hidden Markov models, HAMs, are not used in PANTHER. Pfam is available at 4 locations around the world. Which of the following is not one of them? A. UK. B. Sweden. C. US. D. Japan. The correct answer is Japan. What is the source of protein structures in SCOP and CATH? A. UniProt. B. Protein Data Bank. C. Ensemble. D. InterPro. The correct answer is Protein Data Bank.